Good afternoon, everyone. Thank you for joining us for the FHCC Connect Science Seminar Series. Regarding housekeeping, if you have any questions, please leave them in the Q&A section and not in the chat. It gives me great pleasure to introduce Dr. Richard White, a physician scientist currently at the University of Oxford. Dr. White obtained his MD PhD degree from Albany Medical College in New York. Subsequently, he completed his residency in internal medicine at Yale New Haven Hospital, where he also served as chief medical resident. He then pursued a clinical fellowship in medical oncology at Dana-Farber and MGH, followed by a postdoctoral fellowship under Dr. Leonard Zorn at Boston Children's Hospital. Following his training, Dr. White joined the faculty at Memorial Sloan Kettering Center, where he established a successful research program for over a decade. During this time, he made significant contributions to our understanding of melanoma developmental programs. Recently, Dr. White relocated his laboratory to the University of Oxford, where he also holds the position of Assistant Director of Graduate Studies in the Nuffield Department of Medicine. Dr. White's research focuses on the impact of developmental programs on cancer progression and how the tumor microenvironment can alter these programs. He combines zebrafish and human pluripotent stem cell models of cancer in his research. Dr. White is held in high regard by the scientific community. He is an elected fellow of the American Society of Clinical Investigation, president of Zebrafish Disease Modeling Society, and member of Society for Melanoma Research. Throughout his academic career, he has received numerous prestigious awards, including most recently the Young Investigator Award from the Society of Melanoma Research and the Mentor and Teacher of the Year Award from Weill Cornell Medical College. Today, Dr. White will be sharing his research on melanoma sulfate. Thank you very much, Dr. White. The floor is yours now.
Great. All right. Well, thank you for having me. All right. Great. It's nice to virtually be back at Dana-Farber since my training there, and to tell you a little bit about what my lab has been doing, including some work we recently published and some unpublished work in where we're going now. And I'll be happy to take questions at the end as well. So I'm going to talk today broadly about an idea that we've been investigating for a number of years, something we call oncogenic competence, and I'll explain what that means in a moment. And really looking at this from the inside and from the outside of the tumor cell as well. So I'm giving a talk. So we started this by thinking about this question that I think many of you are familiar with, which is, why are oncogenic mutations only sometimes transforming? And so this is work from the Sanger Institute, Mike Stratton's group, where a number of years ago, they had done a study where they took eyelids from healthy donors and did DNA sequencing on the skin from the eyelids. So these were people who were getting cosmetic surgery. And the result of this was actually pretty surprising, because what it showed is that seemingly oncogenic mutations in genes like, for example, RAS, Notch, P53, are actually surprisingly common, right? That you can have lots of mutations that would classically associate with cancer in seemingly normal tissue. And since that time, there's been a lot of other studies that have shown that genes which we strongly associate with cancer development aren't really transforming in many, many contexts. Another example of this is the fact that oncogenic mutations themselves are extremely lineage specific. So in this data here, this is data from the CBio portal from Sloan Kettering. You can see that genes like BRAF, when they're mutated at V600E or V600K, are really only transforming in a surprisingly small number of lineages, despite the ubiquitous nature of BRAF, right? So we see it obviously in thyroid cancer and melanoma, and then it trails off very fast after that. The one that was surprising to me is KRAS, which I tend to think of as a sledgehammer of an oncogene. Again, very high prevalence in pancreatic cancer and other GI cancers, and really trails off after that, and almost unheard of in melanoma, although it does occur at extremely low frequency. So that gave us a clue that lineage is at least one part of what makes a mutation oncogenic or not. And this was a question that we found really interesting in understanding the mechanisms of this. And the tumor I'll talk about today is melanoma, which I've spent most of my time investigating. I don't need to introduce melanoma here. We know that melanomas arise from pigmented cells called melanocytes, or their neural crest progenitors, and work from my lab. And it's amongst the most heterogeneous of tumors. And this is true at both genetic as well as epigenetic levels, as I'll talk about in a moment. And so in thinking about these questions of what makes a mutation able to form a tumor, melanoma is actually an extremely good model for this because there is so much genetic heterogeneity in these tumors. So in thinking broadly about this idea of oncogenic transformation, I think it's clear from work from my lab and many, many other labs that transformation depends on both cell intrinsic as well as environmental interactions. So work from my lab, for example, has shown that melanocytes can exist along a developmental trajectory from neural crest cells, which are quite undifferentiated. They can undergo a number of epigenetic or microenvironmental shifts to become melanocytes or pigmented cells, and mutations can occur in either of those populations. So even within one tumor, we have both neural crest cells and melanocyte cells. They're all of the same lineage, but obviously in different differentiation states. And each of those tumor states can then really interact with the vast number of cells in the tumor microenvironment. And these include cells like adipocytes in melanoma, a cell my lab has done work on, but also many other cells like fibroblasts, lymphocytes, glial cells if they're in the brain, and keratinocytes, which I'm going to talk a little bit about later on. And really deciphering the nature of the way in which the heterogeneous tumor cell populations interact with the heterogeneous microenvironment is really the major question my lab is trying to address. Melanoma, again, is an excellent model for this because if you just look at a histology of, this is a stage 1 melanoma, this is an early melanoma that's still confined to the skin. And we can see here, I don't know if my pointer is working, the melanomas initiate at the dermal epidermal junction up here, and those early purple cells up here are entirely surrounded by keratinocytes, the predominant cell in the skin microenvironment. As the tumors grow deeper into the skin, they're almost primarily surrounded by immune cells and fibroblasts, and as they get into the subcutaneous tissue, they're now completely surrounded by a different microenvironmental cell, in this case, adipocytes. And really trying to understand how all the different microenvironmental cells that these tumors encounter along this very tiny trajectory, right? This is just a few millimeters down, I think is a real technical challenge. But what I think we're finding is that each of those interactions plays very specific roles at different stages of tumorigenesis. And that's really what we're trying to dissect. So when I was a postdoc at Children's, I decided to join a zebrafish laboratory. So this is the lab of Len Zahn, who some of you may know. So he's an oncologist at Dana-Farber who developed the zebrafish as a model initially for blood development, and then when I joined, for cancer development. And there are some really unique features that made the zebrafish very attractive for me when I joined the lab. And I'm just going to sort of highlight two things that I make it particularly good. The first is that we've optimized lots of technologies for doing rapid non-germline transgenesis. So in traditional mouse transgenesis or knockout studies, you typically pass through the germline, and that's actually a huge bottleneck because you have to breed lots of animals that are not informative and genotype all of those animals. And actually you get a very small number of animals that are informative. And instead, one of the things that my lab has done is figure out ways to directly introduce transgenes into the tissue of interest through electroporation, through a technique called TEAS. So this lets us manipulate many, many genes at once and go directly to the somatic tissue that we're interested in. The second technology, what I developed as a postdoc, is high-resolution imaging by creating this transparent fish called Casper. So on the top here, this is still a zebrafish, but we've engineered it so that way it has no pigment cells at baseline, and then we can mosaically add back those animals. And in this movie that I'm showing here, you can see the tumor starts here and then very rapidly begins to spread to other parts to the animal and then eventually metastasizes to the very anterior of the animal. And with different types of microscopes, we can also do very detailed single cell and subcellular imaging in these animals. So when I think about the zebrafish, I really consider it to be useful for rapid genetic modeling and high resolution in vivo imaging. And those are the two technologies. And we use iPS cells and other things to sort of complement what we can do in the zebrafish, but this is our core model. As I mentioned, not everything can be done in the zebrafish, and we do, of course, use cell lines for certain things. Cancer cell lines are powerful, but the problem is in studying things like tumor initiation, they're actually not great for that because they're obviously long transform. And in collaboration with Lorenz Studer's lab, we've developed a number of systems where we can take human embryonic stem cells or iPS cells, engineer a variety of different genetic lesions into them like BRAF or P16, and then differentiate them into very well-defined stages like neural crest or melanoblast or melanocyte stages. And so I think bringing these two models together has given us a lot of insight into these early steps of tumor initiation. So a lot of our interest in this question about DNA alterations and their ability to transform started actually with this fish we created when I was still a postdoc. So this is a transgenic fish in which the wild type fish is shown on the left here. This is a P53 mutant. This is an animal expressing BRAF V600E under a melanocyte specific promoter MITF. And then this is the combined BRAF P53. And what was striking about these animals was not that they got tumors, but actually that they got only one tumor in general. So this is an animal in which we transformed thousands of melanocytes, millions potentially, but let's say thousands. And yet the tumor generally develops a single fish in highly stereotyped locations. We subsequently went on and did DNA sequencing of these tumors, a panel of 53 of these tumors, and there were no recurrent additional genetic drivers. And so that led us on a nearly 10-year path to try and understand why this is, right? Why are these genetic mutations sometimes sufficient to give rise to tumors, but usually not in the context of this animal? And as often is the case, this led to a whole series of studies, and I'm just going to summarize a number of years of work in thinking about this question. And what we eventually discovered was that the cells that were capable of ultimately giving rise to the tumor in the presence of those genetic lesions had two very unique characteristics. The first is that it expressed very high levels of neural crest transcription factors like SOX10. So this is a TF that keeps the cell in a relatively undifferentiated state. But that on its own was not sufficient. It also needed a binding partner that binds to SOX10 and regulates chromatin accessibility at the SOX10 loci. And that's a gene called ATAD2. And there are other chromatin factors that can do this as well. And so what this led to a model of is that in order for a DNA mutation to be transforming, or a set of DNA mutations to be transforming, you need indeed oncogenic mutations. You need lineage-specific transcription factors, but you also need the chromatin factors that bind to those transcription factors and allow them to then express those programs. And I think one of the things that emerged from this is that at least in melanoma, and I'm fairly sure this is true in other cancers as well, there probably is not a single cell of origin. So depending on the particular combination of these three factors, that will determine the competence of any given cell to respond to those mutations and become a tumor. And so that's why we phrase this idea oncogenic competence, meaning that neural crest cells are particularly competent to form cancer. Mature melanocytes are relatively incompetent, but can be coaxed to do so. And so this is the type of thing that we've been trying to understand for quite a while. But that eventually led us back to the question that I addressed earlier. So that helped us to understand the cell state and the epigenetic mechanism that determine competence. But what it actually never told us is, why do the tumors form where they do? As I had mentioned, in the BRAF model, these tumors tend to develop in highly stereotyped locations, often on the anterior or on the dorsum of the fish, and sometimes in other locations, but very often in these locations. And so that got us really interested in this concept of anatomic positioning of the tumors. Like, why do they appear where they do? So it was right around this time that an MD PhD from Cornell joined my lab and became interested in this question. And he started by thinking about the fact that human melanomas have very distinct anatomic tumor types. So cutaneous melanoma or superficial spreading melanoma, what we think of as the commonplace melanoma, develops on the trunk or the back, or sometimes on the face. So these are called cutaneous or body melanomas. But then there's a very distinct type of melanoma called acral melanoma, which only appears on the hands and feet exclusively. And so that was a real opportunity for us to think about this question of why do the tumors appear where they do? Because clinically, those are such different tumors. And perhaps we could then mechanistically dissect the contribution of this anatomic positioning to help explain this. And so when Josh joined the lab, what he decided to do was to take a series of melanomas and profile cutaneous versus acral melanoma. And we were fortunate at the time that there were a number of groups around the world who were also interested in this question of profiling these two anatomically very different types of tumors. And so this I'm showing here is data from the MSK impact sequencing panel, which is a panel of several hundred genes in all these patients undergoing DNA sequencing of their tumors. And from the impact panel, we get point mutations, but we also get copy number alterations. And what Josh did, as shown on the y-axis here, is he calculated the log2 ratio of DNA lesions seen in acral tumors versus cutaneous melanoma. And the first thing that popped out when we looked at the data was that if you look at the cutaneous melanomas, they are dominated by point mutations, shown in black, things like BRAF and NRAS and P53 and NF1. In contrast, the acral tumors almost never have point mutations. These are dominated almost entirely by copy number amplifications. So these are genes like CRACL and GAP2 and CDK4 and MDM2. And so right off the bat, we knew that at the DNA level, these two anatomically different tumors were quite different and had different mutational spectrum and copy number alterations. So we wanted to know what that means. So to assess that, what Josh did was to use the rapid non-germline genetic techniques that I mentioned earlier to build zebrafish models where he was able to test many of the genes that came out of the MSK impact sequencing. So on the left here, this is a control fish with mosaic rescue of melanocytes, and you can see those are, just relatively normal melanocytes. On the right here, he rebuilt our original cutaneous model, which is BRAF and P53, and again, those animals developed tumors in the same locations. And then in the middle are fish in which he expressed a variety of the different genes that we found in the acral tumors. And these included genes like CRACL, GAP2, loss of NF1, and TERT. And similar to what happened in the cutaneous model, all the fish developed melanoma. But what was striking about it was that the fish with the acral genes, especially CRACL, developed tumors primarily in the fins of the fish and not on the body of the fish. And you can see one example here shown in the red arrow, where this is the fin of the fish, and you can see a tumor is developing right in the fins. This animal actually has two tumors. This is a second tumor in the fin of the fish. And the cutaneous fish developed these big anterior dorsal tumors. And we quantified this and showed that this is an extremely reproducible phenotype. So what do fins have to do with the origins of acral melanoma? So fins are really interesting for those of us who are into this kind of thing, because through a number of studies, there's been a lot of work in connecting the evolution of tetrapod limbs, our hands and feet ultimately, to the origins of fins in fish. I think the most dramatic example of this is work from Neil Shubin and other colleagues who began a journey to try and actually understand the origins of how did fish first walk on land. And it had been hypothesized for a number of years in the evolution literature that there had to be a transition species from fish that were obviously swimming in the water to the first animals that walked on land that eventually became tetrapods. And what was hypothesized is that there must be a fossil out there that has proto-limbs, where the fins had converted into small limbs. Indeed, eventually they identified this fossil called Tiktaalik, and now there have actually been a number of other fossils identified with these proto-limbs. And you can see in this artist rendition, these are animals that still have fins, but clearly have gained a lot of the bony structures that give rise to the limbs, and specifically the hands and feet. So that was very compelling for us because it suggested that there might be an evolutionary relationship between the fins of the fish and the hands and feet of tetrapods and humans. So what's the connection? So to get at this, what Josh did was he took our fish and he dissected off the skin from either the body where the cutaneous melanomas develop versus the fins where the aqueous melanomas develop. And then he used he fact sorted the cells and did both bulk and single cell RNA sequencing on the melanocytes that occur in the body and the melanocytes that occur in the fin. He also sequenced the microenvironment, and I'll get to that a little bit later. And what was really striking about this was that if you look at the pathways that are altered in just regular melanocytes from fin versus body, even in the absence of mutations, the main gene signature that's different between these is a posterior Hox gene signature. So the acral melanocytes express posterior Hox genes like HOXC13 and HOXA13, whereas the body melanocytes express more anterior Hox genes like HOXB6 and HOXB7. And so these are genes that are well known during embryonic development to pattern the AP axis in the embryo. What this data suggested is that melanocytes and indeed other cells in the skin actually retain this developmental history and continue to express low levels of these Hox patterning genes in an anatomic way that makes sense from embryonic development. And so we wanted to understand whether this transcriptional program of Hox genes was somehow important in the pathogenesis of acral melanoma. And Josh did a number of experiments to get at this, and I'm just going to show one for the sake of time. But I think the most elegant experiment was where he took our zebrafish that overexpress crackle, where they get these excess fin melanocytes and ultimately fin melanoma. And then he created a CRISPR vector in which he could selectively knock out either an anterior Hox gene, HOXB7, in the melanocytes, or the posterior Hox genes, HOX13 in this case. And then what he did was he measured whether crackle was still able to induce this excess melanocyte hyperproliferation in the tail fin. And what you can see in the middle here, so this is a control where all these little dots out here are excess melanocytes in the fins of the fish. You can see it here. When you knock out the anterior Hox gene, that doesn't really change very much. But when you knock out HOX13 in its most extreme form, you get a complete loss of the crackle-induced phenotype, suggesting indeed that there's a relationship between a DNA alteration in crackle and the transcriptional program that's inherent to these tail fin melanocytes, notably by HOX13. So what is HOX13 driving to promote these tumors? So we did HOX13 ChIP-seq on fin tissues, including fin melanocytes as well as the entire fin of the fish. We also compared it to mouse limb bud data. There was very good publicly available data for that. And we then analyzed what loci are targeted by HOX13 in these limbs and fins. And what came out of this was primarily genes involved in the IGF receptor signaling pathway. Now, IGF is obviously involved in many things, but why this was very interesting to us is that it's been known from the developmental literature for a long time that IGF proteins are especially important in posterior development of the animal. So it affects other parts of the animal, but the tail fin of the fish is incredibly sensitive to IGF. Moreover, the other thing that was interesting to us is that crackle is an adapter protein. And the way Crackle works is to transduce signals through the RTK pathway, such as IGF. And so this was compelling to us because it led to the hypothesis that Crackle was interacting with this HOX13 IGF pathway, and it was that synergy that was promoting the tumors specifically in the tail fin of the fish. And so to test this, Josh then wanted to ask, well, if we gave the animals Crackle and the melanocytes, but then abrogated this HOX13 IGF signaling pathway, would that actually reduce tumors in the fin? So again, he used our transgenic system, created animals that had Crackle so they get excess tail fin tumors, and then inserted an additional gene, which was a dominant negative IRS2, and then asked what happens. And you can see here on the right, this is what's called a ternary plot. So this is a way of looking at anatomic position of the tumor. So you can see a body versus a head versus a fin. So a tumor that's in the middle is promiscuous, can go anywhere. The apical tumors are primarily in the fins. But when we overexpress the dominant negative IRS2, now we lose these tail fin tumors, suggesting that the hypothesis that this HOX13 IGF axis was indeed synergizing with Crackle because it's an adapter of that pathway. And so that has led us into thinking a lot more about this question of anatomic site. And the model that we came up with is that BRAF is actually a fairly promiscuous oncogene, whereas Crackle and other apical genes that we discovered very much depend on their intrinsic transcriptional program, in this case, a HOX13 transcriptional program. And we think that in part explains why the tumors appear where they do, because of this synergy between a DNA alteration and this RNA transcriptional program that's pre-existing in the program. We're also very curious about whether this applies to other tumors, how often you get a sort of a relationship between a genetic lesion and the underlying positional gene program. There's some evidence in pancreas cancers and colon cancers that this might be the case as well. And I'm going to return to this question of anatomy in a moment. So when we finished this work, there were a number of people in the lab, obviously were following Josh's story along the way and began thinking about this question more broadly of anatomy. And, you know, in the original seed and soil hypothesis of Paget in metastasis, you know, I think we're all fairly familiar with the idea that anatomic position and the local microenvironment in each of those different anatomic positions plays an important role, certainly in metastatic seeding. But we began to think about whether anatomic position in primary tumor formation might actually have a role in tumor initiation, right? So different parts of the animal and the microenvironment in each of those sites might actually be a very instructive signal for shaping this idea of competence, right? Whether a cell is going to form a tumor or not. And you can envision that the primary environment of different anatomic sites of the skin is wildly different than the lung and wildly different than, for example, the liver. So we thought a lot about different ways of asking this question. But the problem that I think should be apparent to most of you is that the TME space is vast, right? You know, there's many, many different cells, as I mentioned earlier, that could be influencing tumor initiation in this context. Even within the skin, there are many different cells that could be influencing this. And I think this is a real challenge for the field because it's very difficult to assign functionality to these interactions. Because the number of potential interactions is so large. And so one of the questions that we have been trying to address over the past couple of years is how do we begin to think about this problem in a functional way and come up with ways of testing these interactions in vivo? And I'm going to start by just talking about some of the work that we've been doing on really, really early melanoma initiation and keratinocytes, which are the primary cell in the skin microenvironment that surround the melanocytes. So this is a melanocyte here, and you can see the number of keratinocytes is vast. And that's ignoring all of the other cells that are here, but we had to start somewhere. And I think what we realized is that we needed a good technical tool to look at these interactions. And essentially what we wanted to develop was a fate mapping system for cell-cell communication. And for those of you who might work in this area, this has actually been a fairly rapidly growing area. There's a number of different tools out there to try and fate map these interactions. So there's one called GPaton from Monty Winslow's lab. There are other ones called mCherry niche, which is a very nice system. But we were attracted to this system that was developed a number of years ago by Jakob and Riemann's lab, which is a Krelok-based system. And the reason why we like this system is because, unlike many of the other methods, which will record transient interactions between the cells, a Krelok-based approach will give us a permanent view of those interactions. And so the way this system works is that you have a donor cell that has a very specific form of Cre recombinase where they've added on a microRNA sequence to it that fates that Cre recombinase for secretion in extracellular vesicles. So that's the donor cell. The recipient cell will then have a Flox reporter, for example, a DS-RED followed by a stop cassette, and then a GFP reporter gene, such that any recipient cell that takes up Cre recombinase will permanently switch, in this case, from green to red. We thought this is a very elegant system for our type of question to look at intercellular communication between tumor and microenvironment. So this is the system that we adopted for a particular question. I'm not going to go through all the details of how we built this, but I'll just simplify this to say we built transgenic zebrafish, in this case, that are expressing BRAF under the melanocyte promoter, MITF. And then those melanocytes will simultaneously then also express this exportable or secreted form of Cre recombinase. So that's our donor cell. The recipient cell in this case are keratinocytes, in which we have a KRT4 promoter driving a Flox black Z, really just as a stop cassette. And then if it receives Cre from the melanocyte, it will switch to being red and should be permanently marked by this. And so that was sort of the system. And we've also now done this for a variety of other oncogenes like Crackle and things like that, but I'm not going to talk about those today. I'm just going to talk about BRAF. And so this is what it looks like. It's a very, very clean system with a very, very low noise and leakiness in the system. So this is an individual zebrafish. You can see, and I'm zooming in on the body of the zebrafish. This is an individual melanocyte, which we false colored yellow here. And this is an individual keratinocyte, which is in direct communication with this melanocyte that has the Cre and also has BRAF and has P53 knocked out. And I think this is really beautiful, right? Because we can actually isolate these individual keratinocytes that are in direct communication with the melanoma cell. There are many, many other keratinocytes in the animal, but it's really only the ones that are in direct communication with these nascent melanoma cells that undergo this switching process. And this gives us an opportunity now to try and understand what's different about these keratinocytes versus all the other keratinocytes that are scattered throughout the skin. So there's a lot of different ways of approaching this question. But before we delved into the mechanism, we wanted to first ask whether this relatively small number of keratinocytes were actually important for tumorigenesis, because it's a Krelok system. There could be leakiness or noisiness. So we wanted a functional test of this. And so the way Mohita Tagore, the postdoc in my lab who did this, is she re-engineered this Cre system in which the melanocytes will have Cre, and they'll have BRAF and all the other things. But what she changed is the Flox recipient cassette. So what she did is she engineered it such the KRT4 promoter has a GFP followed by a stop, so it will switch to red. But at the same time, when it switches from green to red, it will then also express a cell ablation cassette, in this case, the diphtheria toxin. And then what she can measure is the number of tumor-initiating cells that occur from this. So the idea being that if these keratinocytes are important for the survival of these melanoma cells, ablating those keratinocytes should somehow then affect the ability of those melanoma cells to survive. And that's exactly what she sees. So here you could look at the number of nascent melanoma cells, so cells that are capable of becoming a melanoma. In the control animals, we get about 20 per animal. And in the ablated animals, which we've gotten rid of the switch keratinocyte, it essentially drops to 0. So this was very, very cool for us because it sort of told us that even though there are not that many of these keratinocytes, they must be doing something quite special for the tumor. So the question was what? So the way that Mohita chose to undertake this was to use a genetic screening, a chemical genetic screening approach to try and address this question. So the way she set this experiment up was she took melanoma cells and keratinocytes in culture that had the exact same switching cassettes I just talked about. So these are BRAF melanoma cells that express this secreted form of CRE, and the keratinocytes in the culture express the FLOX GFP to RFP reporter. And really the question we were trying to get at here was a mechanistic question. If we could apply a broad library of chemicals, could we then understand what might be the involved mechanism in this switching? Because we had shown that the switching is important for tumorigenesis, but we had really no idea what mediated the switching itself. So Mohita completed the screen, and this was the result of the screen. So on the x-axis here, these are different compounds that came out of the screen. On the y-axis here is the fold increase in switching compared to the DMSO control. So as I showed earlier, the control animals only have a few switched keratinocytes. But in the presence of these different chemicals, this massively goes up. So this one chemical here, it's a nearly 20-fold increase in switching efficiency. And then a number of other chemicals as well. But what was striking about the screen is of the top 10 chemicals that emerged from this screen, 5 out of the top 10 all hit the same exact signaling pathway. And they all converged on the GABA-A receptor. And in doing screens, I think it's sort of a truth of screens that if you get 50% of your top hits hitting the same signaling pathway, even though the chemicals are quite diverse, that's probably biologically meaningful. And so that's what Mohita has decided to follow up. There are other hits from the screen that we've done a little bit of work on, but I'll only talk about the GABA-A aspect of this today. So GABA, as most of you probably remember, is an inhibitory neurotransmitter typically produced in presynaptic neurons in the brain. GABA itself is synthesized by 2 enzymes called GAD1 and GAD2. GABA is then packaged into synaptic vesicles, is released into the extracellular space. It then binds to GABA receptors present on the postsynaptic neuron. That leads to a conformational change in the channel that leads to an influx of chloride ions and therefore hyperpolarizes the postsynaptic neuron. So that's why it's considered an inhibitory neurotransmitter, because it tends to lead to hyperpolarization of the neuron. And what Mohita's data gave us an idea that might be happening was this question of whether melanomas and keratinocytes might be forming synapse-like structures. Not synapses per se, because I think that's really a term that probably should be reserved for for neuronal cell types, but certainly a intercellular communication or junction that's mediated by GABA, and specifically the GABA-A receptor. So we use the term synapse here somewhat carefully in quotes, because I think it shares some characteristics, but certainly not all. So I think that there were reasons to think that this might be a synapse-like structure, and that really has to do with the morphology of a melanoma cell. So if you look at normal melanocytes here on the left, or melanocytes that have been transformed with BRAF, they have this very beautiful dendritic structure. This has actually been known for many years, and in fact, in the melanocyte field, we've called these dendrites for a very long time, for no particular reason other than the fact that they look neuronal and they kind of look like dendrites. And so I think it was a reasonable hypothesis that GABA might be mediating a type of cell-cell communication somewhat reminiscent of how neurons communicate through these processes, like these dendrite-like processes. And so the first thing we wanted to test on a functional level was whether GABA was actually important as a mediator of tumor initiation. So in this experiment, what Mohita did was to engineer the fish in the exact same way as before. But in this case, she engineered in an animal in which the keratinocytes now express a dominant negative GABA-A receptor. So this is a gene called GABARG2. And so everything else is the same here, except that the keratinocytes should no longer be able to respond to GABA. And then what she measured on the y-axis here is the number of initiated melanoma cells that have BRAF and CRE and all the other things in there. And what you can see, again, in the control animals, we get somewhere around 10 to 20 initiated melanoma cells in these animals. But in the keratinocytes that are expressing the GABA, the dominant negative GABA-A receptor, now we get a very significant reduction in this. It's not a complete reduction the way we saw with the cell ablation, and that probably is because cell ablation is obviously a much bigger thing and things other than GABA involved. But I think this is a very interesting and compelling result that GABA is explaining at least part of the communication between these two cells. So we wanted to know whether this was true in human melanoma as well. So we analyzed data initially from the Wistar melanoma cell lines. This is a very nice data set where they did expression profiling on normal melanocytes, primary melanoma, and metastatic melanoma. And what you could see is that melanocytes essentially have no expression of the GABA synthesizing enzymes, GAD1 and GAD2, whereas the melanomas do. But I think even more compelling in our IPS system that I described earlier, if we take cells that are competent to form tumors, so neural crest cells and melanoblasts, and we turn on BRAF, we see this really massive induction of GAD1 and GAD2. Whereas the melanocytes themselves, similar to what was found here, even when we turn on BRAF, really cannot induce the GABA synthesizing enzyme, suggesting that it's something quite specific to the melanoma state, specifically related to BRAF, that seems to induce a high-level expression of GAD1 and GAD2, and subsequently GABA itself. So we next wanted to ask whether these special GABAergic keratinocytes were actually tumorigenic. So the idea being that we had said earlier, if we ablate them, that seems to get rid of the tumor-initiating capacity. We wanted to ask in established melanoma, if we inhibit GABA signaling or this communication between these keratinocytes and melanoma cells, would that reduce the growth of these melanoma cells? And so in this experiment, what we did was we took normal keratinocytes, what we call parental keratinocytes, or the switch keratinocytes, which are GABAergic and have high levels of GABA receptor, and then measure the proliferation, in this case, of A375 melanoma cells, and clearly see an increase in proliferation, suggesting that these GABAergic keratinocytes are indeed promoting the growth of not only initiated melanoma cells, but also established melanoma cell lines like A375s as well. We did some further analysis on human samples to ensure that we also saw this in actual human tissue biopsies. So we collaborated with a pathology group at Sloan Kettering, and we got a tissue microarray of 6 normal skin samples, as well as 6 primary melanomas. And as most of you probably know, getting primary melanomas for immunofluorescence is quite difficult, because those are usually used for diagnosis. But we were able to get 6 of each. And then we stained those for KRT17 as a marker of keratinocytes, S100 as a marker of melanoma cells, Geffen is a marker of the GABA receptor, and then you can see the merged image on the right. And what's very striking here, similar to our cell line data, so in the normal tissue, we see lots of keratinocytes, no melanoma cells, no Geffen staining in the melanoma cells because there's no GABA, presumably in normal melanocytes. But in the presence of these early initiated tumors, you can see we have lots of keratinocytes. These are the melanoma cells shown in yellow here, and these now are the Geffen staining in the keratinocytes. So clearly the interaction between the melanoma cells and the keratinocytes is what upregulates the expression of the Geffen receptor or the GABA-A receptor cluster. So this gave us confidence then that there was this interaction not only in zebrafish, but also in human cells and in human tissues. So we wanted to do further high-resolution imaging to understand what the nature of the GABA receptor is on the keratinocytes, because GABA receptors, if they're acting, again, not synapse exactly, but synaptic-like in a certain sense, we wanted to see whether the Geffen staining, which marks the GABA receptor, had characteristics similar to what was seen in neuronal synapses. And so in this high-resolution image here, the blue cells are the keratinocytes, the white cells are the melanoma cells, and the yellow is the staining for the GABA receptor. And what's quite amazing about this is that really you only see the GABA receptor clustering at these very, very focal points and not along the entire keratinocyte membrane. And if we zoom in and do higher resolution reconstruction, what you can see here, excuse me, is that you can see that the melanoma cells in yellow here, the keratinocytes in purple here, they really only contact each other at very, very focal points. So again, I think consistent with the idea that these share some characteristics of synapses, meaning that they're focally adherent to each other rather than everywhere, but certainly not all features. We further went on and did electron microscopy and saw quite a similar thing. So this was the high resolution imaging showing these focal points, and in electron microscopy, again, we can see a keratinocyte here, a melanoma cell here with a melanosome, and you get this very, very focal point of contact between the melanoma cell and the keratinocyte. But ultimately, if we're hypothesizing that GABAergic signaling is happening between melanoma cells and keratinocytes, we had to show this using electrophysiology. Because as I mentioned earlier, GABA is an inhibitory neurotransmitter. It induces hyperpolarization of the recipient cell. And so to test this, what we did is we used this technology called a multi-electron array, which is basically a chip that has thousands of spikes sticking out of it that can measure electrical activity, and each metal spike will go into a single cell. So each black dot here represents an electrical signal. And then what we did is we tested whether the melanoma cells were communicating with the parental keratinocytes, which are low in GABA, versus the switch keratinocytes, which are high in GABA. And I think this is easiest to see on this movie, which shows the spike plots. So each of these blue lines here is an individual spike, meaning that there's an electrical communication between the melanoma cell and the keratinocyte. And so with the parental keratinocytes, which are low in GABA, there's quite a lot of electrical signaling going on, as evidenced by the blue spikes. And in the switch keratinocytes, which are high in GABA, we see this great diminution of this signal. So there's still spikes occurring, but at a much lower frequency, and this is entirely consistent with a hyperpolarizing phenomenon. We showed in other data that this is dependent on chloride in the media, but I'm not going to show that data now. And so this then led us to this question: if this hypothesis that GABA is somehow mediating this connection between this population of keratinocytes and this population of melanoma cells, we wanted to then test whether GABA was necessary and sufficient for tumor initiation. And so in this experiment, what Mohita did was to create animals that have BRAF and P53, just like we've done before, but then in the melanocytes, further overexpressed GAD1 and GAD2, which are the GABA synthesizing enzymes. And what you can see here, and I think the plot on the bottom right is the most instructive, is something we had actually almost never seen in the fish before, which is that in the control animals, as I discussed earlier, they develop one tumor in stereotyped locations. But when we overexpress GAD1 and GAD2, now we get animals with extremely multifocal tumors, up to 6 tumors per animal. And this is really something we had never seen before, suggesting that just having excess GABA coming from the melanoma cells and interacting with the keratinocytes makes the entire microenvironment far more permissive for tumorigenesis, even given the same exact genetic lesions. And we also did the loss of function experiments in which we knocked out the GABA synthesizing enzymes. And I think this is quite dramatic. This is using this electroporation technique I talked about. And you can see here, these animals initiate the tumor very well in the control setting. And when we put in guide RNAs against the various GABA synthesizing enzymes in the melanocytes only, now we get an almost complete abrogation of tumor initiation in these animals. And so I think this really tells us that GABA, mediated by GAD1 synthesis in the melanoma cell, is both necessary and sufficient for tumorigenesis. I don't have time to go through all the ways that these GABA-ergic keratinocytes promote the tumor. I had mentioned earlier that we know from functional studies they increase proliferation. And I'll just summarize by saying that the GABA-ergic keratinocytes release many different growth factors into the extracellular space, including very notably LIF. And when LIF then binds to the LIF receptors on these melanoma cells, that induces a growth advantage to those cells. And if we block the LIF receptor, we can block this system. And so the model that we have right now is that a melanoma cell that acquires a BRAF mutation, a P53, or perhaps a PTEN mutation, will upregulate GABA synthesizing enzymes like GAD1. That will cause a release of GABA into the extracellular space. That will bind to the GABA receptors on the keratinocytes. That induces a chloride flux, hyperpolarizes the cell, and that releases a number of different growth factors, including LIF, which then feed back on the melanoma cell. And so we tend to think of GABA now as a tuning factor. It's not that other things can initiate tumors, but what I think our evidence suggests is that GABA and this interaction with the keratinocytes makes the cell more permissive to become a tumor when this special type of keratinocyte is nearby. And obviously, we're interested in other things from the keratinocytes as well. And so I'm just going to summarize the way where we've been thinking about this question of why DNA mutations are not often transforming. And I think we've done a lot of work on the developmental state of the cell, and I briefly mentioned this. We know that anatomic position is really important, like what oncogene is able to form a tumor depends on where that cell is. And I think we're very excited about this idea of using this microenvironmental fate mapping system for understanding how different cells in the microenvironment can then push those cells over the edge to become tumors. And I showed an example of keratinocytes that have GABA receptors, but now we can apply this fate mapping system to virtually any cell in the microenvironment to try to understand the interaction. So we're doing this with the adipocytes, and we're doing this with fibroblasts and other cells, and we're excited to see where this takes us. And finally, I'm just going to end with a thought, actually, regarding this idea of anatomic positioning, and because it's so important that there are cell intrinsic programs in these anatomies and then microenvironmental programs. And now in Oxford, we have a number of programs looking at how are cells in different sites specialized? Why is a fin different than the body? Why do they become specialized? We don't understand why you have melanocytes in the palms of your hands or under your fingernails. They're not UV responsive, that's probably not their primary evolution function, but we don't know why you have melanocytes in these sun-shielded areas. And we're trying to understand that. And then, of course, does this apply to other cancers as well? And there are lots of really interesting animals for trying to get at this question of why melanocytes in different parts of the body and different microenvironments have very, very different functions. So we're really excited to understand this. And I just want to thank two people in particular, Mohita Tagore, who was a postdoc in my lab, who did the work on GABA signaling with the keratinocytes, and Josh Weiss, who did the work on the anatomic positioning, and many other collaborators. So I'll stop there and happy to take some questions.
Thank you very much for that fantastic talk, Dr. White. Great amount of work. We have a question from Patrick about the fish model. Do most fish develop one tumor, or are there many fish that do not develop tumors at all?
Yeah, that's a great question. In the BRAF model, every single fish develops a tumor without exception, which is actually quite interesting in a lot of ways. In the crackle model, 70 to 80% of the fish develop tumors, and there's a small number that don't. And so we don't exactly understand the reasons for that, but those are the numbers we have.
Thinking about different melanoma subpopulations, which one do you think is producing GABA? Are there known connections with BRAF inhibitor resistance and changes in GABA production in melanoma? How much do you think GABA contributes to other cells in the tumor microenvironment? This question comes from Joanne.
All right, so I'm gonna give two answers because they're kind of two intertwined questions. The best data we have so far is from our IPS system, where when you turn BRAF specifically on, GAD1 and GAD2 just go skyrocket. We're currently mining other data sets right now to see, is this a general phenomena of oncogenesis, meaning could NRAS do this, could KIT do this, could Crackle do this? And I don't have the answer to it yet, but we know at minimum, if you don't have BRAF, even in a neural crest population, which is super susceptible to tumorigenesis, you don't have GAD1 and GAD2. So that's the first answer. The drug resistance question is really interesting. I don't think we have very firm data. My lab has not done a lot with drug resistance. We would be extremely excited if somebody who does work in this area wanted to look at this question with us, because we just, honestly, in the fish, we don't have great tools to look at this. Of course, we could look at, human cell lines, but we haven't extensively looked. And then there actually was a third question. Mohita is going to start her lab hopefully in the next year or so, and that's actually what she wants to look at is when GABA is released from the melanoma cell, what other cells is it interacting with besides keratinocytes? And there's actually very interesting data about interaction with immune cells, that immune cells often express GABA receptors. And so she's in the middle of doing experiments to try and look at what is the repertoire of cells in the TME that respond to GABA that comes from the melanoma cell. It's a fascinating question. We don't have the answer, but that's what she's looking at.
That's exciting. So we have a question from Gustavo. Amazing talk, really beautiful. Is GAD1 enough for mature GABA production? There are two more questions that I got.
Yeah, I'll address this. We have done studies where we only added GAD1 or GAD2. I think it is sufficient, but pretty weak. It seems like when both are present, it's a much more potent effect. But I think GAD1 by itself, the data we have, it can be sufficient on its own.
GAD1 and GAD2 activity may require glutamine. By any chance, is there any cell type that may fuel GABA pathway with glutamine or even L-glutamic acid?
Can you repeat the last part of the question one more time? I heard the first part, just the very last part.
Yeah. By any chance, is there any cell type that may fuel GABA pathway with glutamine or even L-glutamic acid?
Oh, God, that is such an awesome question. Obviously, I don't have the answer. Yeah, I know we've thought about this question of where's the glutamine coming from, right? And is it coming from another cell in the microenvironment? I have no insight into this, but God, that would be something amazing. Because you could imagine it could actually be far more complicated than we've shown, right? Where you have some cell is donating glutamine to the melanoma cell, that then produces GABA, and then GABA goes to the keratinocyte or to an immune cell. So it could be a far more complex ecosystem than I think we had appreciated. Yeah, we don't know where the glutamine is coming from.
Finally, any additional direct stain for GABA or any vesicular transport, such as VGAT?
Yeah, we've tried a whole ton. The only antibody that we got to work, and even this antibody was hugely problematic, was this Geferin antibody. The reason we haven't done others is that as I mentioned, primary biopsies are really rare. So we have very, very few samples to work with. So once we got Geferin to be good enough, that's all we could stain for, because we simply don't have more tumors than that. Even getting those 6 was really hard.
Udochi asks, in the BRAF model, do the fish develop tumors in up to 2 anatomical positions?
Yes. BRAF tumors, they actually develop in 3 anatomic positions. And again, we don't exactly understand why. They're promiscuous in the sense that they develop these anterior head tumors, these dorsal tumors, and some fin tumors. I didn't want to imply that BRAF never gives rise to fin tumors. They do, but they don't preferentially give rise to fin tumors, as opposed to crackle, which is obviously gives preferential to the fin. I just heard a talk recently about somebody who engineered an animal with HRAS, which is an exceptionally rare tumor mutation in melanoma. It's like 1 or 2% of patients. And that remarkably only gives rise to fin tumors. So it's not just the RAS pathway. So BRAF gives rise to pretty promiscuous tumors, but HRAS for some reason seems heavily fated towards the fin.
Chang asks, does GAD1 have gain of function mutation with BRAF or P53 in melanoma in clinical?
Not that we can really, not that we ever convinced ourselves. We did look, we looked in CBio and TCGA, but nothing that ever convinced us that these were truly co-occurring events.
Balaji asks, does GABA signaling have a role in cell fate specification, crosstalk with Hox genes?
Yeah, great question. So Mohita is one of the ways that Mohita is trying to address this, which I think would get at your question, is we never actually looked at what is the role of this in normal development, right? We showed data that GAD1 and GAD2, at least in a static picture, are very low in normal melanocytes. But we don't actually know in reality how GAD1 and GAD2 might be dynamically expressed over the course of melanocyte development. So she's doing experiments now to engineer the Cree into normal melanocytes with the fate mapping system in recipient cells to try and understand, is there a window period in which having GAD in the melanocyte either changes melanoma cell fate if we overexpress it, melanocyte cell fate, or changes its interactions during development. So those are ongoing experiments. It's an excellent question. We know at baseline, there's not much GAD, but if you overexpress it, what happens? So
Matthias asks, could you talk about the LZTR1 gene and its synergy with the CRKL gene on acral melanomas? Congrats for the presentation.
Oh, thanks. So Ruth Halaban had this really nice paper about LZTR1, and that gene is often co-amplified, and we found that as well. We actually built fish with it. And I think it's an equally important gene in acral melanoma. The only reason we actually didn't do more with it was because we were very surprised that when we built fish that had crackle alone, they got acral melanomas all by itself without any other cooperating DNA alterations. This is in contrast to cell culture. If you take crackle and stick it in melanocyte in cell culture, the cell will synapse. And we reproduce, the Halaban lab saw the same exact thing. So that suggests that there's something very special about crackle in the in vivo situation compared to the in vitro system. So that's why we actually haven't extensively looked at the other cooperating events, because we were so struck by this difference between crackle in vivo versus in vitro.
Last question for the day. Do diabetes people have more incidence on acral melanoma?
Yeah, not to our knowledge. As you know, because diabetes is such a heterogeneous disease, and melanoma by numbers especially advanced melanoma, is a rare enough disease. It's actually hard making a statistically confident statement about this. Certainly there's nothing we've seen in the literature that, is solid about this. But it's a good question. I just don't know that we're powered enough to answer the question.
Thank you very much, Dr. White. This was a fantastic talk, and thanks everyone who joined us today.
Yeah, it was great being back and seeing everyone. So have a good rest of the day.
Thank you, you too, and have a good weekend to everyone.
Yeah, thank you for having me.